All right, so today we're going to continue our discussion of eukaryotic cells. Okay? And last time we did a lot of me talking about the structure of eukaryotic cells, and today we're going to be doing some more practice for exam 2. So it's going to be a guided study session, if you want to think of it that way. All right, so today we're going to be progressing toward being able to meet these learning objectives, defining the key role of evolution as it applies to microbiology, okay? Identify microbial structures and connect the structures to their functions, analyze and describe the impact of microorganisms, okay? All right, before we begin, any questions? All right. Let us move on. If you think of some questions, let me know. I'm happy to answer them. All right. So the first thing we're going to do is we're going to practice drawing and labeling a eukaryotic cell. Okay. So what I'm going to do is I'm going to go to the whiteboard. And the plan is, without consulting your notes, okay, because this is an excellent study technique. By forcing yourself to remember, okay, excuse me, by forcing yourself to remember, it's kind of like a mental workout. Okay, if you make yourself mentally sweat before you look up the answers, you remember them for longer. Okay, and so because this is a practice session, it's okay if you get it wrong. That's part of study. Okay. Sometimes you get the wrong answer, you go in and you correct it, and you remember the right answer for longer. Okay. All right. So here's what I want you to do. I want you to talk me through drawing a eukaryotic cell. Okay. And we're going to label it while I draw it.
Draw a circle.
Okay, so I'll draw a circle, a big circle or a little circle?
A big one.
A big one. Okay, I'm going to draw a big circle. Okay, very good. So let's go ahead and label this guy. Okay, what is this big circle we've drawn?
The membrane.
Very good. It's the membrane. I'm going to go ahead and put cell membrane. We could label it plasma membrane. Okay. All right. What is its function? What is its job for the eukaryotic cell?
It keeps things from going in and out that shouldn't.
Very good. Okay. I'm going to abbreviate that, going in and out. Okay. All right. Okay, so, but we do need things to go in and out. What helps things go in and out? That's technically a part of the cell membrane, but I tend to do them separate.
That phospholipids?
Yes, phospholipids, yes, are a part of the cell membrane. Okay, let's go ahead and put phospholipids here. Okay, so just so that we know, let's go ahead and blow up a part of this. So how would you draw a phospholipid membrane?
Where she's bell?
Yeah.
Sorry, what?
And the shoulder.
Yeah, how would we draw this phospholipid bilayer? Oh, I just gave it away. How do you draw a bilayer?
It goes through.
There are some things that go through, but are they?
Oh, the
phospholipid?
bilayer goes around the top of it.
It does. Yeah, very good. But how would we draw it? If I were to take my cartoon here of the yellow line and I were to blow it up,
Wouldn't it be like a circle and then two legs underneath it?
very good. Circle and two legs. Okay. What about on this other side?
It would be a mirrored version, having the two legs touching.
Very good. Okay. Good job. Okay, so that's my phospholipid bilayer. Okay. All right. So what are the things that go through? Let's go ahead and draw one here. Let me go ahead and get the phospholipid here. Bilayer. Okay, it's a bilayer because we've got two mirrored phospholipid layers. Okay, so what is here that is a part of the cell membrane that allows things to go in and out in a controlled fashion? Okay.
It's a protein.
It is a protein, yes. And we call it a what kind of protein because it goes clear through the membrane.
Transport protein.
It is a transport protein, yes. And we have another name for it too. I'm going to go ahead and put transport protein here. Okay.
Can I ask a question, back up a little bit? So
Sure.
the phospholipid bilayer, that's part of the membrane that doesn't lay on top or bottom of it, but it's integrated into the membrane.
It is the membrane. I
Are
don't
they
know if that helps.
all are they all that, or do only some of them contain those?
So all membranes have this phospholipid bilayer.
Okay, then
Yeah.
why do they say membrane and not phospholipid bilayer?
Because they want to be confusing. No, I'm kidding. I think phospholipid bilayer describes just the lipid portion of the membrane. Okay. The cell membrane is composed of the phospholipid bilayer, but it also has transporter proteins embedded in it. Okay. So like I could draw it like this if I'm doing the cartoon version, and this is the blown up part. Okay. I also have proteins that are stuck on it that kind of stick in. Okay. I've got others that are kind of stuck in it and they stick out. Okay.
So it's a composite of or a component of the membrane.
Yes, yes. The phospholipid bilayer is a component of the membrane, but it's the part that makes up the biggest part of it.
Okay, thank you.
Yeah, so the cell membrane is composed of phospholipids, and proteins. Okay.
Okay, thank you.
Oh, you're quite welcome. And sorry, sometimes I use them interchangeably at the beginning, which is understandably confusing. I will have to stop doing that. Okay, so we've got our cell membrane here. Okay, I'm not going to draw any more proteins just because it takes too long. Okay. All right, so what else do we have in a eukaryotic cell?
A nucleus?
A nucleus. Okay, and it's composed of what? And how do I draw it?
It's got chromosomes in it.
It does have chromosomes in it. Let's go ahead and put the chromosomes in here. And I'm going to draw them in cartoon form. Okay. Here. Now, in reality, they're all coiled up with the proteins, but for the sake of the cartoon, we're going to go ahead and draw them this way. Okay, and then I'm going to put some proteins to kind of hold them together. All right, so this is my chromosome. And just for the, or chromosomes, just for the sake of repetition, what are they made out of?
DNA,
DNA and,
protein, I don't know.
oh, no, very good, you did it right. Proteins. Okay, now, what do the chromosomes do? What are their function in the cell? What's their job? Now, this one's a little harder. I'm going to make you all mentally sweat for a little bit.
Chromosome, oh, sorry.
No, you go.
Chromosomes contain the genetic information that essentially tells what different proteins do and how to build them and stuff like that.
Very good. Hey, very good. That is the job of the DNA and the chromosomes. Okay, that's the genetic information, and the proteins protect, okay, the genetic information. Okay, so in a eukaryotic cell, the chromosomes are in which structure? We said it before, but for the sake of repetition.
I forgot the question.
Oh, let me go ahead and repeat it. So the chromosomes are inside of what structure in a eukaryotic cell?
The nucleus.
The nucleus. Very good. Okay, and this circle that I've drawn, okay, we call that the nuclear membrane, but this entire thing is the nucleus. Okay, the nucleus has the chromosomes, and it has a nuclear membrane.
Is the nuclear membrane the same as the nuclear envelope?
Yeah. Yep, tell you what, let's go ahead and I'll cross that out and I'll put envelope. It depending upon the textbook you use, they will call it either a nuclear membrane or a nuclear envelope. It means the same thing, but let's not be confusing. We'll use the term this book uses. Okay, so what is the nuclear envelope? Because it's a membrane, is composed of what? Chromosomes are made up of DNA and proteins. Okay, the nuclear envelope is made up of what?
Wouldn't it also be proteins?
It would be proteins and
A phospholipid bilayer.
very good, phospholipid bilayer.
Isn't the nucleus also double membrane bound?
It is double membrane, yeah. Yeah, I tend not to draw it that way just because, you know, the cell's getting crowded
Cool.
already.
Yeah,
But-
super.
Yeah, but let's go ahead and I'll go ahead and I will indicate that it is a double membrane by, we'll put a line here And that's kind of the outer membrane or the outer portion of the envelope. We don't want to call it an outer membrane because that's on the outside of gram-negative cells. And then we've got another membrane here. Whoops, boy, I am just not sticking to I'm not staying in the lines today here. There we go. Okay. And we have pores. Okay. We have big old channels. Okay. That allow things to come in and out of the nucleus. And we call those nuclear pores. Okay.
You
And
call
that's
them
how
what?
things get in and out. Sorry, what?
I'm sorry, you call them what?
Nuclear pores.
Thank you.
So they're kind of like the transporter proteins that let things in and out through the cell membrane, through the plasma membrane. Okay. But they're in the nuclear envelope, which is a double membrane. So when we get to the chapter on genetics, mRNAs come in and out. Well, they come out. Okay, nucleotides that are made out here go in. Okay, we have information from the cell that goes in, information from the nucleus comes out, and they all go through the nuclear pores. Okay, what's another structure? What's another structure that is inside of a eukaryotic cell?
Ribosomes.
Chromosomes. Oh, let's see. Yes, we have chromosomes that are right here. So yes, very good.
Ribosomes.
Oh, I'm sorry, I misheard you. Yes. Okay, let's get some ribosomes going here. Okay. And we'll draw one that hasn't clamped onto an mRNA yet. Okay, so what size are these guys?
$70s.
You know what? You were close.
Maybe.
Eukaryotic cells are a little bigger than prokaryotic cells, so they are
80.
very good. 80s. Now we do have some 70s ribosomes in a eukaryotic cell, but where do we find them? What structure do we find them?
The mitochondria.
Mitochondria. Yes. Okay, so let's go ahead and draw a mitochondrium. I'm only going to draw one. Okay, and it has, let's go ahead and put its 70s ribosomes in here. We'll just put 2 for the sake of space. Okay, and why do mitochondria have 70s ribosomes? They used to be
They evolved from the prokaryotes.
very good. Yes, they evolved from the prokaryotes. Good job. Okay, so ribosomes, whether they're in the cytoplasm, the ADS ribosomes, or the 70s ribosomes that are in the mitochondria, you know what? I'm going to color that in so it doesn't look like an 8. We'll color all of them in. Okay, what is their job? What is their job in the cytoplasm of the eukaryotic cell or in the matrix of a mitochondrium?
They take RNA that was
Mhm.
copied from DNA and create enzymes and proteins.
Very good. Okay, so they read the mRNA to make proteins, which include enzymes, structural proteins, transporter proteins, at least the start of them. Okay, you name it, they make it. Good job. Okay, so what are ribosomes made of? Let's go ahead and put what they're made of down here just for the sake of it, but they both make proteins and they're both composed of the same things. What does the ribo stand for? Okay, we've got RNA in them. Okay, it's a cousin of the DNA over here. Okay. And then they also have what? What holds the RNA in place? The macromolecule that can do just about any job.
Protein.
Proteins. Good job. Okay, so since we have a mitochondrium drawn here, what's the mitochondrium's job? What does it do? What is its function in the eukaryotic cell?
It's the powerhouse of the cell.
Very good. It is the, whoops, helps if it's spelled right. Ah,
So doesn't it make ATP?
it does. Very good. Yes. Okay. So it makes ATP. Good job. Okay. All right. What's another structure?
You have
Oh.
the endoplasmic reticulum.
Very good. We have the endoplasmic reticulum. Okay. So we have them in two flavors. What are they?
Smooth and rough.
Very good. Smooth and rough. And how do you tell the difference between the two?
The rough ones have ribosomes on them.
Very good. Rough ones have ribosomes. Okay. So we're going to go ahead and dot this one with ribosomes. Okay. And we're going to go ahead and label this the rough endoplasmic reticulum. Okay. Then we have the smooth endoplasmic reticulum. Okay. What do these guys do? I'm going to write the functions down here just because we're getting a little crowded here. Okay, so the smooth endoplasmic reticulum and the rough endoplasmic reticulum, what do they do? What's their job in the cell?
The smooth endoplasmic reticulum is where synthesis of lipids, fats, and
You got a good start.
the membranes as well, right? Phospholipid
Right.
bilayers get synthesized there.
Yep.
Anything that basically isn't proteins.
Very good. Yes. So where do we make the proteins? Okay, I think I heard an answer, but not quite sure.
Can you please say again what the smooth ones do?
Okay, so the smooth ones make lipids, fats, phospholipids, okay, and anything that is not a protein, okay? So lipids are not proteins. What are the two other macromolecules that are not lipids?
Would amino acid?
I'm sorry, I didn't quite get that.
Would amino acids be one?
Yep, amino acids would definitely be one. And amino acids make what?
They eventually make proteins.
They do eventually make proteins. But we take amino acids, we send them out here. Okay.
Carbohydrates are also another.
Oh, good. Carbohydrates. Okay. And if we have the capability, we also make nucleotides there. All right, so with the rough endoplasmic reticulum, it's studded with ribosomes. Ribosomes make what? So the rough endoplasmic reticulum also makes
Protein?
protein. Very good. Okay. So these amino acids get made here, they get shipped here, okay, they get shipped out here to the cytoplasm for the free ribosomes to hook them together to make proteins. Okay. All right, you guys are doing a good job. All right, let's do one more. Let's do one more organelle.
Would it be the Golgi apparatus?
Very good. Yes, it would be the Golgi apparatus. Okay. Boy, that's a sad looking Golgi. Okay, but there are stacks. There's stacks like stacks of pancakes or stacks of pita. Okay, what does the Golgi do? What's its job?
Doesn't it activate enzymes?
It does. It does activate enzymes to do what? In other words, you're on the right track. Keep going.
It breaks to break things down.
The other direction.
It creates.
Yes. Okay, so it modifies. Okay, everything that was made here and here. Okay, so it modifies macromolecules. So, like, it would activate enzymes to put proteins and carbohydrates together to make the glycoproteins. It would take lipids and proteins and put them together to make lipoproteins. And it would also take proteins and move things around and change this and change that to get them to their final form. Okay?
Yeah, forgive me. I was mixing
no
up
worries.
with lysosomes.
No problem. And you know what? It is really easy to do that, which is why we're practicing. So let's go ahead and put a lysosome over here. Okay, so go ahead and repeat what lysosomes do. And then I'll finish writing over here.
They'll generally encompass and break down structures into their base components.
Yep. So they break down structures and macromolecules. Okay, good job. And over here we modify proteins, etc. Okay. All right, good job, everybody. So that was kind of tough. That was tough, but we got a good mental workout. Okay. So this is something that you can do on your own or in a study group. Okay. And it's hard. It is hard, but as you do it, it gets easier. So it's kind of like when you're in the gym and you're trying to use a new piece of equipment or bench press more weight. Okay. It's hard until you build up your muscles, you build up those neural connections in your brain, and then it becomes easier, and then we move on to something hard. Because you know, you gotta keep moving. Okay? All right. So, like I said, if you were going, I got no idea what's going on here. And that's just fine. That is just fine because at the beginning, especially since we just barely learned about this the last time we met, you know, it's still something that you're struggling to keep in your long-term memory. Okay, but you're going to remember it after today. You're going to remember this for longer. Okay, you will start to forget it, so you do it again. Okay, we can't go to the gym just once and keep our muscles. We have to keep working them. Okay? All right, any questions about this study technique? Okay, this is one of my favorite study techniques because I'm more of a visual kinetic learner. So drawing things out and labeling them really works for me. If I were more of an auditory person, then saying what they look like with their jobs, where they are, that might work better for me. Okay. All right, let us go to, there we go. Let's go back to the PowerPoint. Now that we've done the visual kinetic stuff, let's go ahead and move on to more auditory. Okay, so we're going to pull up the polls. And we're going to do some polls here. Okay, so here we're going to do something where we're looking for things that are similar between prokaryotic and eukaryotic cells. Okay, so this is an understanding style question. We're comparing and contrasting. So which of the following is a similarity or it's the same between prokaryotic and eukaryotic cells? Once again, if you're going, I got no idea. Okay, remember this is a study session. We're going to give you a mental workout. Okay, we've got about 50%. Good job. Okay, there we go. We're almost, there we go. We just broke 70%. Ooh, we broke 80%. Let's see if we can get to 90. Oh, we're at 90%. Two more and we got 100%. We're kind of stalled at 90%. All right, for the sake of time, I'm going to end the poll and I'm going to share the results. Okay. The favorite was cytoplasmic membrane composed of phospholipid bilayer with ester linkages. Okay. Now you're probably going, ester who? This goes back to that table that's in the week 5 lecture folder that has the characteristics of bacteria versus archaea versus eukaryotic cells. Okay. All right, and B is correct. Okay. Now, if I said ether linkages, that's unique to archaea. Okay. So ester, okay, that is bacteria and let's see, and eukaryotes. So if I had been more accurate, okay, I should have put, okay, bacteria and eukaryotes. Okay, so this is mostly right, but not completely right. Okay. All right, so the next favorite one is presence of mitochondria. Okay. So that one, we see mitochondria only in eukaryotic cells. Okay. So if you pick that one, don't feel bad. Okay. We're learning, it's just at the beginning. Okay. So circular chromosome composed of DNA and scaffolding protein. Okay, prokaryotic cells and eukaryotic cells both have a chromosome composed of DNA and scaffolding proteins. But in eukaryotes, they're in a line, they're linear. And in both prokaryotic cells, bacteria and archaea, they're circular. Okay? And then the last one, ribosomes are the same shape and size. Actually, we've got different shapes and different sizes between the three domains. Okay? So archaea and bacteria both have 70S, but they're different shapes. Okay? Eukaryotic cells have 80S ribosomes, but the shape is pretty close to that of archaea. Okay? And nobody picked sexual reproduction. You're right, only eukaryotic cells engage in sexual reproduction that we know of. Later on, we're going to talk about how bacteria and archaea exchange DNA, but it's not sexual reproduction. All right, so actually there was no right answer, but this one was the closest answer to being correct. Okay, I kind of pulled a fast one on you this time. Okay, let's move on to the next one. This next one does actually have a right answer. Okay, which of the following is a difference between prokaryotic and eukaryotic cells? Hey, we're almost to 50%. And we're at 85%. Let's see if we can get to 90. I think we're stalled at 85%. We'll wait a little bit longer. Okay, let's go ahead and end the poll. And we're going to share the results. Okay, so most of you picked ADS ribosomes. That is correct. Yes, eukaryotic cells have ADS ribosomes. Prokaryotic cells have 70S ribosomes. So that is a difference. Okay. All right, the next favorite answer is the presence of a cell wall. Okay. If you picked that one, you were almost right. Okay. Some eukaryotic cells have cell walls. Okay. And most prokaryotic cells have cell walls. Okay. But you probably picked that one because animal cells, we're animals, and we do not have cell walls. Okay. So good job on remembering that animals do not have cell walls in their cells. Okay. But fungi, plants, algae, they're eukaryotes and they have a cell wall. Okay. So this was a almost correct, incorrect answer, which is pretty good for this stage in learning the material. Okay. The next one is chromosomes composed of DNA. Okay. Actually, both prokaryotic and eukaryotic cells, okay, have chromosomes composed of DNA. Okay, now you may have picked it because I didn't put the protein scaffolding. Okay. So, you know, I can see why you picked that. Okay. And asexual cell division. Okay, if you pick that one, you know, that one was actually showed good reasoning because in the previous question, okay, eukaryotic cells undergo sexual cell division. We form gametes, okay, that combine to form a new eukaryotic cell. Okay, we don't see that in bacteria. We don't see that in archaea, but actually we see asexual cell division in both prokaryotic and eukaryotic cells. Okay, Even at this moment in your body, your skin cells are dividing asexually. They're making clones of themselves, identical twins, to replace the skin that's dying and shedding. Okay? Same thing with the rest of your body. Okay? So eukaryotic and prokaryotic cells both undergo asexual cell division. It's just eukaryotic cells are the only ones that we know of that make gametes, okay, and undergo sexual reproduction. Okay? All right, good job. They both have flagella. Good job. Okay, let's do another one. Okay, this one's a little harder. Okay. What characteristics of chloroplasts and mitochondria resolved the debate as to if they are prokaryotes that have become internal symbiotes of eukaryotic cells? Okay, so what sealed the debate? You know what? I'm going to end the poll on this one because I don't have a correct answer again. This one was not on purpose. Oh, I'm gonna go ahead. You know what? No, I don't like this question. Never mind. What I intended to do was to say genetic relatedness instead of linear chromosomes. My apologies. I intended B to be the correct answer, but I didn't give you the correct answer. So my apologies. I'll have to fix that for tomorrow's class. These are all things that led to us coming up with an S being capital O, or capital U, because you know, we argued back and forth. The fact that they have double membranes as if they had been engulfed, okay, that was a good clue. Eukaryotic cells have linear chromosomes and mitochondria have circular chromosomes. Okay. Eukaryotic cells have 80S ribosomes, mitochondria have 70S ribosomes. Okay. Cytoplasmic membranes with ether linkages, they both have that, but that didn't lead to us having the discussion. Okay. But it was actually the genetic sequence that sealed the deal. So my apologies, I did not write this question very well. Let's move on to the next one. Okay, this one's better. This one is better. Okay, so fungi are more similar to animals like helminths, worms, than they are to protozoa in that, okay, this one I actually have a correct answer. Yeah, this one's a little harder. Okay, we're almost to 50%. We're almost to 75%. There we go. Ooh, at 80%, let's see if we can get to 90%. Okay, I think we're stalled at 80%. Okay, so the favorite answers were they both have unicellular or multicellular species, and they both have cells that undergo asexual division, mitosis, or productions of gametes, meiosis. Okay. All right. So, hey, let's look at this one first. Okay. Fungi definitely have unicellular species. What do we call those? If I'm a fungus and I'm single cell, what am I called?
Mold.
Almost. There are some mold that are microscopic, but they're always multicellular. I know you don't think of them as that. Yeah, yes, yeast. Good job. Okay. And then what's another multicellular version of fungi? There's mold, and then there's that's the fuzzy stuff that grows on fruit and bread. And then we have the stuff we put on pizzas that are delicious.
Mushrooms.
Mushrooms. Yes, very good. Okay, so with animals, okay, we actually do not have single-celled species of animals. Okay, all animals start out as a single cell, but that zygote, that fertilized egg and sperm, very quickly divide to form two cells. Okay, so you know, this one was good reasoning. You were remembering the fungi are multicellular and multicellular and unicellular. Okay, but animals are always multicellular. Okay, so if you pick that one, don't feel bad. Okay, it's a we're in the process of learning, and you showed some good reasoning. Okay, so fungi, okay, and animals both have cells that undergo asexual division, mitosis. Okay, that's how we get more, okay, of the non-reproductive cells, and they both produce gametes. Okay, okay, very good job. Okay, so this one, for the purposes of this class, okay, is a correct answer. Although later on, when you learn more about protozoa, you will find that there's weirdness going on with protozoa. Mostly they do mitosis. Mostly they do asexual reproduction. But there are some protozoa that do sexual reproduction, but not through meiosis. Okay. Yeah, they do some funky weird stuff, at least so far as I know. Okay. All right, so this one is also a correct answer. Okay. Fungi and animals are always non-photosynthetic. Okay. We animals cannot photosynthesize. We've got to eat plants. We've got to eat other animals. Okay. And fungi do the same thing. They just don't take bites out of them like we animals do. Okay. They release enzymes out into the environment and break stuff down and then suck in the broken down nutrients. Okay. But they're both non-photosynthetic. So good job, whoever picked D. Okay. E, if you picked D, don't worry. Okay, we're still in the process of learning. Fungi have cell walls, animals do not. Protozoans don't have true cell walls. Okay. Okay, this one was harder. This one was harder. Okay. It's good to practice with these types of compare and contrast style questions. Okay. So you can make your own compare and contrast kind of activity to study. Okay. So what I would do is I would set up a table and fill out what I could remember. Okay, these things are similar, these things are different, and then go back to the book and then compare and see which ones you got right, which ones you got wrong. Fix the ones you got wrong, fill in the things you forgot, okay, and then start over. Okay. All right, let's do another one. Okay, many protozoa are similar to some bacteria in that. Okay. So now we're comparing across this chapter and the previous chapter 3 when we talked about bacteria? Protozoa are eukaryotic, bacteria are prokaryotic. How are they similar? We just broke 50%. We just broke 75%. We're at 80. See if we can get to 90. Ooh, we're at 85. Just need one more portion. Oh, there we go. We got 90. See if we can get 95. Ooh, 95. Let's see if we can get 100. Okay, let's share results. Oh, nope, I didn't want to do that. Okay. Okay, so this one you can tell it was harder. Okay, when you take information across 2 different chapters, it's harder. Okay. So if it turns out that you picked the wrong answer, that's perfectly okay. We're studying, we're practicing today. Okay. All right. So the favorite answer, okay, was that both have circular chromosomes composed of DNA. Okay. Now this is a partially true statement. So if you picked it, that's perfectly okay. You showed some good reasoning. You remembered things that were similar. Okay. But which part of this is incorrect?
Would it be circular?
Very good. Circular. Which ones have circular chromosomes? And it's okay if you don't remember.
Bacteria.
Bacteria, very good. And protozoa have what shape?
Linear.
chromosome? Linear, good job. Repetition. Repetition is a good thing for studying. Okay, the next favorite answer, okay, was both have cell walls composed of peptidoglycan. Okay. Okay, so protozoa have structures that are similar to cell walls. Those who study protozoa say that they're not true cell walls. I personally, I don't know why, but they say they're not true cell walls. So this would not be correct because protozoa do not have cell walls, whereas bacteria do. Okay, and bacteria have cell walls that are composed of
Peptidoglycan.
peptidoglycan. Good job. Okay, so we have a tie between A and D. Okay, so let's go ahead and take A first, just because it's first on the alphabet. Okay, so do bacteria have unicellular species?
Yes.
Very good. Yes. So do bacteria have multicellular species?
Yes.
You know what? And not that we know of, but that is perfectly okay because we're just learning this. So far of the bacteria that we have found, they're all unicellular. Okay. Protozoa, so far as we've found, they are all unicellular. Okay. So They are similar in that they're both unicellular, okay? But I made it incorrect by tossing in this multicellular part, okay? All right, let's look at D, okay? They are both always non-photosynthetic, okay? Now this one's a tough one, okay? Because we haven't talked about it. But we do have protozoa that are green and they photosynthesize, okay? We have bacteria that are green and photosynthesize, okay? But not all of them do. Not all of the protozoa and not all of the bacteria photosynthesize, okay? So this one is a little too broad. If I put that they both have non-photosynthetic and photosynthetic species, that would have been a correct answer, okay? Okay, so the other one that tied, okay, E, actually this is the correct answer, okay? Both many protozoa and some bacteria have a resistant resting stage, okay? Does anybody remember the term we use for this resistant, hard to kill stage of bacteria that some bacteria produce?
Endospore.
Very good, endospores. Good job. Okay, who remembers the term that we use for a resistant resting stage of a protozoa? Yeah, this one you may not remember because, you know, we're just covering this chapter this week. Haven't had enough time to have repetitions.
Can I give it a shot? I might get it
Sure.
wrong though. I want
Oh,
to say
feel free.
mycoplasma.
Oh, you know what? You're close. Yeah, mycoplasma do have a resting stage, okay, but they're bacteria. Okay, so good job giving it a shot. Yeah, perfectly okay to be wrong in class. Anybody else want to give it a shot? Starts with a C. And we use this term to describe structures that we humans get that we generally need to have removed. You're going, I have no idea now.
Cyst.
Yes, very good. Yes, protozoa, some protozoa, many protozoa form cysts. It enables them to survive dry periods. So it's like endospores and bacteria. Okay. And when we get to the chapter on disinfectants and sterilization methods, endospores and cysts are both very hard to kill. Yeah. Because, you know, if they're already dried out, you can't dry them out and kill them. They're resistant to heat. Yeah. Yes, Brandon.
I just had a question going back to B. I wanted to confirm, so with circular and linear chromosomes, it doesn't matter if they're multicellular or unicellular, just if they're prokaryotic or eukaryotic?
Yes. Yes. If you are eukaryotic, it doesn't matter if you are multicellular or single cell. Yeah, you always have a linear chromosome. Okay, you notice I put that in quotes. We're not going to worry about the one or two exceptions that are just weird. Bacteria always have a circular chromosome, and they're always single celled.
All right, thank you.
Quite welcome. Okay, now having said that, the exception is mitochondria. Okay. But I would specify that I was talking about mitochondria for that. Okay, other questions? Okay, let's move on. Hey, here's another one. Okay, this one is, we're going to talk about helminths, worms. Ew. Let's say in this scenario, this would actually be an application style question. Okay. Where I give you a scenario and you take what you know and you apply it to this new situation. Okay. So in this situation, you're a school nurse in an elementary school. Recently, there's been an outbreak of pinworm infections in the students at your school. Ew. Okay. Which of the following would you do to prevent further spread of the infection at your school? Okay. All right, so I'm going to relaunch the poll. So for this one, what you do is you take what you know about the life cycle of pinworms, okay, and think about how would you stop that?
I'd prefer to burn it down at that point.
I don't blame you. I didn't give that option, did I? I should have. Hey, by the way, there's more than one answer I would accept. Okay, we're at 76%. Actually, because there's, I would, yeah. I'm gonna take out the all of you, but then we don't get to have a good discussion. Okay, we're at 85%. %. See if we can get to 95. All right, let's go ahead and end the poll, share results. Okay, most of you picked all of the above. Okay. Hey, that's a good answer. We'll say it's a correct answer. Okay. There's just one of them that, you know, might not be something that you could do as a school nurse. Okay. And this one, you could send home notes suggesting that the parents have their children checked and prescribed anti-worm medications. Okay, so that is a correct answer. That would help prevent spread of the infection in the school. I'm just not sure how well you can do that as a school nurse, but sure, this is a correct answer. Okay, what or oh, excuse me, I got D and C confused. Sorry, nobody picked that one, but they had it as part of the all of the above. Okay, so that would prevent spread by taking some anti-worm medications, which would shut down the replication of the worms, okay, which are laying eggs, which are going out in the feces. Okay. Well, all right, let me back up. They don't actually do that. They do lay eggs, but what they do is at night they come and lay the eggs around the anus, which makes it itchy, and so you'll scratch and you get them underneath your fingernails, and then you can re-inoculate yourself, or you can pass it on to other people. I know. Ew. Okay, so we have something to pick. Teach effective hand washing, especially under the nails, to children. Okay, because pinworms, the eggs get on the nails, underneath the nails. That would be very effective. Okay, but you gotta rely on kids washing their hands well. Okay, and it takes us a while to learn how to do that, to get the good fine motor capabilities and practice in washing our hands. Okay, so this is one of the best answers, but let's be realistic. Not all the kids are going to be able to do that. So that's why we have this other option, okay? Treating the kids so that we get rid of the pinworm infections. By the way, pinworm infections are self-limiting, okay? Your immune system, for most of us, will clear that out if given enough time and if you don't keep re-inoculating yourself. Yeah. Yay, instruct the teachers to pass out an ethanol-based hand rub like Purell to all of the students before lunch and recess, okay? That, would probably be pretty effective, especially if you got it underneath your nails. Okay, that would drop the numbers of eggs on the hands. Okay, and disinfecting the restrooms with bleach. That's also a good answer. Okay, because wherever the kids are putting their hands, okay, or spreading the eggs, okay, we're gonna kill them. Okay, good job. You guys did a good job on that hard question. I really should take this one out so that whether we could talk about why each one is a good answer. I'd be forcing you to pick. Okay. Actually, I think this is the last one that I'm going to have you answer. Okay. Okay, and this one talks about free living water protozoa that have been known to cause brain infections. Okay, brain eating amoebas. We're planning a vacation to Hawaii and are planning several swimming adventures. Which of the following would convince you to limit your swimming to the hotel pool? This is another application style question. We're almost to 50%. Good job. We're at 50%. Well, there we go. We broke 75%. Okay, 84%. Almost to 90%. We are at 90% if I round. Oh, there we go. Just need one more person to have 100%. Oh, for the sake of time, I'm gonna go ahead and end the poll. Okay, most people picked pathogenic protozoa are found in warm marine waters. Okay, would that keep you out of the ocean? It'd keep me out of the ocean. Okay, so if you picked either C, D, or E, okay, I would not consider that a correct answer, okay, because one, we have, if we found the protozoa in glacier fed lakes, okay, that would keep me out of swimming in this area, okay. But so far as I know, Hawaii doesn't have glaciers, okay. But if you didn't know that, there are some islands that have in the tropics that have glaciers. Hawaii just isn't one of them. Okay? I wouldn't go swimming in acidic hot springs, and I wouldn't go swimming in bog marshes. I mean, beaches. Ooh, okay. This one, I'm glad nobody picked this one because this is a joke answer. I will toss in joke answers to relieve tension on tests. Okay. So this one is the correct answer. That would keep me swimming in the hotel pool as opposed to swimming in the ocean.
Do you know that I've known two people that have had that? One died and one took years to get better.
Seriously, wow, you're above average. Wow.
They
So how
were
did they get them?
they were it was actually down in Mexico. And they were out surfing.
Oh dear. Yeah, so yeah, we've found brain-eating amoebas anywhere where the water is warm.
Yeah, well, and the problem was it was years and years ago, and it took them a while to figure out what it was. So they were doing all kinds of other things before they realized what was going on, and the brain was just swelling and getting bad. It was terrible for both of them.
Oh dear. Yeah. Oh, that's horrible. Not very many people get brain-eating amoeba, which, like you said, it takes them usually a long time to figure out that's what's going on. With recent cases, because we've had enough cases, there's about one per year in the United States. And considering that we have millions of people living in the United States, your chances of getting it are pretty low. But physicians now know to look for it, especially if you've been exposed to warm water. So yeah, at least with the friend that survived, have they fully recovered or mostly recovered?
You know what? He's still a little bit different, but for the most part, he's back.
That's encouraging. That is encouraging, because yeah, most people die from it. Yeah, so yeah, you're gonna remember all the information on the brain amoebas.
Probably.
Yeah, because you've got a personal connection to it.
Mm-hmm.
If we have a personal connection to a new piece of information, we tend to remember it, because my analogy is, in your brain, you've got shelves. If you have a personal connection to the information, you've already got a shelf labeled and ready to put that information. So.
Well, and the reason I know too, I know you said it's very uncommon. The reason I know two people is that I was very involved in the surfing community for 20 years. And so that would be why I would know more than one. It wasn't just because I happened to have friends who surf. It's because that was a community I was extremely involved in and knew many people.
Right. And so, there's a slightly higher probability of getting a brain-eating amoeba. So, yeah, so knowing two people out of the hundreds of thousands of people who do surfing, yeah, that's about the right average. Yeah, it's about the right average. So, yeah, I don't know, even though it's a low incidence, the thought of having something crawling up your olfactory nerve into your brain and starting to eat your brain cells, I don't know, that's creepy. Yeah, that's creepy. But it is encouraging that your friend who survived has mostly recovered. It tells you how the brain is able to recover from these kinds of injuries. It takes a long time.
Yeah, it did take a long time.
Yeah, it takes a long time, but it is possible. So there's hope. It's nice to have hope. All right, let us move on to what to do for next week. Okay. So as a reminder, I don't have it up here, but as a reminder, exam 1, you have until Saturday to take it. Okay. So make sure you're done before 11:59. And then Sarah, check 2. If you've been working on it, you don't need to do anything. If you haven't been working on it, hopefully you start working on it again at the beginning of this week. You'll get most points. Okay. So I will go in and I will take a look at where you're at, and I'll put those points in. Okay. And because I have a due date associated with it in Blackboard, and because I put the points in after the due date, it's going to show up as late. It's not because you did anything. It's just because I put the points in after the due date. So there'd be no point in putting them in before the due date, because you still have that time to get it done. The biochemistry badge is due not this Friday, but a week from Friday. So continue to work on that. And then next week, we're going to talk about genetics. Okay. Any questions?
Teresa, when you say Sergo check 2, do you just mean the chapter 2, the second little box completed?
Oh, okay. So here's what I mean. For the Sargo check 2, I'm going to check to see how far you are in chapters 1, 2, 3, and 4. Okay. So for chapters 1 and 2, I expect you to be at level 1 or higher. If you're at level 1 or higher, then I'll give you full points for those chapters. For chapter 3, I want you to be at level 0.5 or higher. And then for chapter 4, you just need to have started it. Okay, be at 0.1.
All right, thank you.
Hey, quite welcome. Any other questions?
I had a question. So did you say the exam is you said it was 60 questions and is it timed?
It is timed. I think it's 50 questions and it's worth 75 points.
Okay, thank you.
Oh, you're quite welcome. And yes, you have an hour and 15 minutes in which to take it, 75 minutes. And you do have to take it not a lot. Okay, all the questions. Okay, let's go ahead and call it good for today.